Scientists across the globe are racing to find a way to treat coronavirus. Understanding the human immune response to the virus promises to give experts, officials, and citizens a clearer picture of the pandemic. To identify who has an immune response against the coronavirus, it's important to detect an antibody called Immunoglobulin G, or IgG. IgG antibodies are proteins produced 7 to 14 days after an infection by the immune system in response to an infection and are specific to that particular pathogen. People who have been infected by the coronavirus produce distinct IgG antibodies regardless of whether they had symptoms or not. IgG antibodies bind to the surface of the coronavirus, which has 4 types of proteins: spike, envelope, membrane, and nucleocapsid. The spike protein is the major surface protein that the coronavirus uses to enter human cells. The receptor-binding domain, or RBD, of the spike protein binds strongly to a protein on the surface of human cells called angiotensin-converting enzyme 2, or ACE2. ACE2 is the entry receptor, acting as a doorknob to the human cell. Once the viral membrane fuses with the human cell membrane, the genome of the virus enters the cell and begins infection. It's believed that IgG antibodies that bind to the receptor-binding domain of the spike protein may be key to determining the immune response to the virus. To identify an immune response against SARS-CoV-2, it's important to target the right antibodies. Laboratory studies have shown that antibodies against the RBD of the spike protein are neutralizing in vitro, indicating that they may be a more promising measure of potential immunity when compared to antibodies against other SARS-CoV-2 viral proteins. The ability to identify and assess a patient's immunity to coronavirus has significant implications. We still don't know if these antibodies will protect against coronavirus infection a second time. Researchers can better answer these questions with detailed information about the antibodies produced from infection and how long they persist in the body. IgG antibodies against other viruses have been known to sustain for months to years, depending upon the virus and the individual. This is why Beckman-Coulter's Access SARS-CoV-2 IgG assay detects antibodies to the receptor-binding domain of the spike protein, which has been identified as the most promising target to confer an immune response. High-quality antibody testing offers a chance to identify frontline healthcare providers, patients, and community populations who have potentially developed an immune response to the coronavirus.